Everyone. I just got the recording notice. As Melissa said, it's June, so we have the treat this evening of hearing from our McLeod Scholarship recipients. And it's going to be a double header tonight. The way this is going to go is I'll introduce our first speaker, Michael, and he'll present. And I think for the most part, we'll just try to transition right into Stephanie. I'll do an introduction at that point for Stephanie, and then she'll have the opportunity to present. And we'll do all the Q&A mostly at the end. If there's a really pressing question right after Michael's presentation, maybe we can take that, but I think that'll be a good flow for the night. So without further ado, it's my pleasure to introduce Michael. Michael is conducting his research in the Geoecology Lab here at Cal Poly in San Luis Obispo with our very own board member, Dr. Nishi Rajakaruna. And Michael is investigating a very cool thing, the biodiversity of rock-dwelling lichen communities along a coast to inland gradient of decreasing maritime influence. Such a cool study topic. Michael, a little background on Michael. He has a BA from Pomona College. He has served 2 years in the Peace Corps in Panama. And he has also worked as a consulting biologist, I assume here in California. Michael, you can correct me if that's wrong. He also shared with me a fun fact that after 30 plus years of an outright disinterest in microscopy, his new research subjects have given him a new fondness for unraveling the mysteries of lichen taxonomy under the old microscope. So with that, I'm very pleased to introduce Michael, and go ahead and take it away.
Great, well, thanks Kristen for the introduction. I'm gonna go ahead and try to share my screen. This is always an adventure, but it worked earlier. Can everyone see that okay?
Looks good, Michael.
Great. Okay, so yeah, again, thanks for the introduction. Again, I'm Michael Mulroy. I'm an MS student here at Cal Poly, member of the Geoecology Lab. And my advisor is Dr. Rajakaruna. I'm not sure he's here today yet, but he may be joining us. So my project is looking at lichen biotas of Ultramafic as well as sandstone outcrops along a coast inland gradient. Sorry, I'm losing my voice here. That starts from the coast and goes about 70 kilometers inward. And I'm looking at 8 ultramafic outcrops and 8 sandstone outcrops along this gradient. And so in my photos in this intro slide, I've got an upper photo of an ultramafic outcrop, pretty standard inland ultramafic outcrop, and a lower slide of a sandstone coastal ultramafic outcrop. I've got 2 quad frames here. The right-hand one, sorry, I'm losing my voice. The right-hand one is from the sandstone outcrop, and the left-hand one is from the ultramafic outcrop. And so it just gives you a really good idea of how variable these lichen communities on rocks can be. And so at the moment, I've sampled 12 out of my 16 outcrops. I'm about almost halfway through the lichen sampling. I wouldn't even be that far, except I have an awesome collaborator, Jason Dart, who is hugely helpful in Saxicollis lichen identification. I would say he's probably the guy for that in SLO County. So I just want to start off with a little bit of background. When I'm talking about lichens, what is a lichen? So back in the good old days, we talked about lichens as a fungus and a photosynthetic partner, which was either an alga or a cyanobacterium. It's gotten a little more complicated since then, but I think that's still probably the best way to understand lichens and talk about them, at least in this context. So you have a nice close-up photo of a pretty beautiful lichen, in my opinion, on the left-hand side. And if you took a razor blade, which is what I spend a lot of my time doing now, making a really thin section of that lichen and putting it under a microscope, you can see that there's usually not always, but usually two pretty distinct layers, a fungal layer, and then below that harboring an algal layer, which is the photosynthetic partner in this case, which is basically providing energy that the fungus harvests in exchange for protecting the photosynthetic partner. So I also want to give a little bit of background on one of the substrates I'm looking at, Ultramafic Substrates. Around San Luis Obispo County, this is a pretty common substrate relative to other regions in the United States and also in the world. Here, our ultramafic outcrops are primarily what we call serpentine. And serpentine soils and rocks are really harsh substrates for higher plants. And so CNPS SLO chapter might be pretty aware of serpentine outcrops because they harbor so many rare and endemic plant species. I've got one example of one that hopefully we'll be seeing pretty soon, Calochortus obispoensis. And that's just one of many pretty interesting endemic species to ultramafic substrates. And the reason there's so many ultramafics is because these substrates are really hard to grow on. And so only the really well-adapted species can survive. Serpentine is really low in plant essential nutrients as well as high in metal content, including some really toxic heavy metals to plant life. And so you have these rock outcrops. These are actually just my lichen sampling sites. Two of these photos are, and then there's a third photo from near Figueroa Mountain, another ultramafic sampling or outcrop that I was just curious about. And you can just see from these photos, there's just not a lot of vascular plant cover or diversity there. But if you really looked closely, you can see that a lot of these rocks are just really well covered with lichens. So that brings me to sort of what we know worldwide about ultramafic substrates and lichens. Really, all those interesting features of ultramafic plant communities are absent in ultramafic lichen communities. You can get high or low cover and or biomass in these lichen communities on ultramafics. Species diversity isn't really remarkable compared to other rock substrates. And lastly, and kind of most interestingly, there's really little or no known ultramafic endemism in lichens. So there may be one species where there's solid evidence that it's an ultramafic endemic out in Europe. But even that is sort of tenuous, and all you have to do is find a population off of ultramafic and realize that you just hadn't sampled enough areas to really understand the distribution of the species. And that's really the case for a lot of lichens. And just to illustrate how little lichens care about the harshness of ultramafic substrates, that left-hand photo there is a pretty distinctive and common lichen out here in SLO County, Polycoliona luteominia varbolanderi. And that's growing on the serpentine mineral chrysotile, which is also known as naturally occurring asbestos. And you can actually see the little chrysotile fibers under those red dots, which are the fertile reproductive structures of the lichen. And then this is just one of my quadrat frame of one of my quadrat sites on an ultramafic outcrop with a pretty healthy lichen cover of really brightly colored species in that case. So despite the sort of uninteresting nature of lichen species of ultramafics, there are some interesting patterns in lichen biotas or communities on serpentine. And one of those is that the mix of species on ultramafics is often unique when you compare it to other non-ultramafic substrates in the same area or in the same climatic conditions. And that includes having a mix of silicicolous and calcicolous species within the same rock outcrop often. Silicicolous, is analogous to low pH affinity species, and calcicolous is also analogous to high pH affinity species. So basically these are sort of acidic environment preferring species and more basic environment preferring species. So just narrowing in a little bit to talk about what we know about lichen biotas of ultramafics just within North America. There have been 5 studies on this topic, 2 in California, 1 in coastal Washington, 1 up in Mount Albert Quebec, and one in coastal Maine out on the East Coast. So, and there are a lot of more areas of ultramafic substrates than this in North America. So there's a lot more potential to uncover new species or extend distributions. But this is what we're working with so far. And combined, one sort of project I have for my thesis is just reviewing what we know about ultramafic lichens of North America. And so combining the species list from all of these published studies, we have a species list of 370 species for North America. And this shows some similar characteristics from the worldwide analysis of ultramafic lichens. Low similarity in species composition, a mix of high pH and low pH affinity species within a given site. And really, honestly, taken as a whole, a little bit unremarkable aside from that low pH, high pH affinity mix at a given site. There's one species has been found in all 5 of the studies on ultramafic lichens, Candelariella vitulina, with a photo there. And then another lichen, only a single one has been found at 4 out of the 5 sites. The majority have only been found at 1 or 2 of those sampled locations. All right, so this brings me to my sampling study. So I've divided my sampling effort into coastal, intermediate, and inland zones. And within each zone, I have ultramafic or serpentine sites represented by the green circles, and then the white squares are the sandstone sampling sites. And so within each of those sites, I have quadrat locations that I sample, and I take a % cover for each of those quadrats. So overall, for all my sites, I have 128 quadrats total. And what we do basically is, it's me and Jason Dart, my collaborator, with whom I really couldn't execute this project, to be honest. We take lichen cover class data for each of the morphospecies we identify within the quadrat. And for each of those morphospecies, we go and find a voucher specimen, which we can later bring to lab and identify. And so that'll give us ultimately a pretty robust community data set that we can use to test some hypotheses and compare the similarity of these communities on different rock outcrops. In addition to this, we have geochemical data from each of the sites, as well as climate and topography, which we're going to take into account. And at the quadrat level, we're looking at rock slope aspect and microtopography as well, to see how those relate to the community composition that we find. All right, so we're interested in testing 2 hypotheses here. The first one is that since we have these coastal sites and also these intermediate and inland sites, we're interested in understanding whether maritime factors related to being close to the coast override differences in substrate on lichen communities in coastal areas. And so what our prediction is based on that hypothesis is that our coastal sandstone and ultramafic sites will be compositionally, in terms of the species that occur there, compositionally more similar than inland sandstone and inland ultramafic sites in terms of species composition. And then a second hypothesis that we're interested in testing is that coastal climate and salt deposition factors may create conditions favoring high lichen species richness. In other words, we predict that high species richness, or we would predict the highest species richness along the coast, decreasing further inland along our coast inland gradient. So like I mentioned, we've sampled 12 of our 16 sites so far, and we've identified about half of the lichens on those sites, or voucher specimens from those sites. So I think we have about 500 lichen collections so far. We're over 200 of those identified as species. But in order to evaluate this hypothesis, we really need to finish probably, maybe not hundreds of voucher specimen IDs, but at least a good chunk of those, as well as finish sampling those remaining 4 sites. For the second hypothesis, there is some indication that we have higher coastal species richness. So our highest richness so far among the sites we've sampled are at San Luis Hill, which is just up the coast from Avila Beach. It's within the Diablo Canyonlands property. And then El Chorro Regional Park also has really high species richness. And so just as an example, this is one quadrat, that's north-facing quadrat from El Chorro Regional Park. And that I just checked a little bit earlier, and we had 12 species within that 20 by 20 centimeter quadrat. And that was pretty normal for that site. San Luis Hill and El Chorro Regional Park both had some quadrats where we recorded more than 20 species. So pretty remarkable for such a small little space. There's a lot of hidden biodiversity in these rock outcrops, especially the coastal ones. All right, so this brings me to just some interesting findings. The first is that we have 13 new species records for SLO County. I put new in quotation marks just because what I used for this was the Consortium of North American Lichen Herbaria website, which has specimen records that have been accessioned and inputted into that online herbarium consortium. And it's anyone's guess if someone has already collected this or has it sitting identified and just not accessioned yet. And I know for a fact that Jason Dart, my collaborator, has collected a few of these and knew they were around, but we also happen to collect them in our quadrats. And this really goes to show that there's so much we don't know about these Saxicollis lichen communities in terms of the diversity that they harbor and the species composition, that we could have fairly common or widely distributed species here in SLO County that we just don't know about. And in fact, we found some of those already. All right, in addition to this, in the sites that we've sampled so far, we've got 19 new species to add to the Ultramafic lichen species list for North America. So just as a reminder, there's the 370 taxa previously identified in published studies for North America, and also for the worldwide Ultramafic species list. As of 2018, that sits at about 881 taxa. So we have 19 new Ultramafic lichen records for North America, 13 of which are also worldwide new records. And I just wanted to get more into photos now. Here are some characteristic lichens of SLO County Ultramafics. These range from like really showy and charismatic to like barely visible, even with a hand lens. So Polycoliona ignea falls in that category of really showy, cool looking, often forming big patches on ultramafic rocks, especially in inland locations, but it's also a pretty coastal species. So you can see it really almost anywhere on ultramafics in SLO County, as far as I can tell. And then on the other end of the spectrum, we've got Lichenella stipatula, that upper left corner lichen, which you can barely see with a hand lens. It's pretty nuts, but it's a really dominant and often ubiquitous, not often ubiquitous, it's a pretty characteristic species of the sites that we've sampled. And then we have Candelaria vitellina, which is expected. That's the one that occurs, has occurred on all 5 previously published works on ultramafic lichens. And then one that I'll talk a little bit about later is Toninia submexicana, which is a new county record, but also is at multiple ultramafic sites that we've sampled. So it may be rather common, but it's also a new record for the county, as far as we can tell. And then Polycoliona bolacina, the waxy fire dot lichen, is on the lower left there. This is a really charismatic one. It's kind of got this shiny, waxy look to it, big fertile reproductive structures. And I think Jason Dart took this picture that I'm using here of an apothecium under polarizing filters, which gives it a really neat look. And polarizing filters are used to identify certain compounds that can help you identify lichens. So that's probably why he did that. And it just ended up looking really cool. All right, so just want to wrap up with some notable species that we've found so far. So the first one is a really cool, this is probably the coolest one that we found so far. This is a rare species that had only been known from Southern California and Baja California, as well as the Channel Islands. And we got it up at El Chorro Regional Park. This represents about 140-kilometer northward range extension for this rare species. And who knows how far further north it goes, but it goes a good deal further north than we thought it did. So that one was a really, really neat find. Another one is Rhizoplaka Glaucofana. This one is really charismatic. It's one of the few lichens I would say you could fairly confidently identify with a little bit of practice without having to bust out a microscope. We have a record on an inland ultramafic site that fills about 160-kilometer range gap for this, which is a little surprising since it is so showy and easy to identify. This is endemic to California and Baja California, and incidentally, my advisor, Dr. Roger Karuna, recorded it in San Benito County, also on Ultramafic, in an earlier study. And then I mentioned this species a minute ago, Tonininia submexicana. We have a northernmost California coastal record for this species. But we found it in coastal, intermediate, and inland ultramafic sites. So that's fairly surprising to have a northward range extension along the coast anyway. And also, just checking the chat. Okay, questions, cool. And also to have it on multiple ultramafic outcrops. So really interested to see what other species are actually turned out to be common that we just haven't found in SLO County or haven't been formally recorded yet. Another one I wanted to include, this is outside of SLO County. We recorded this in southern Monterey County. This is a northward range extension for Acarospora robiniae. This is a yellow Acarospora species. And it's not too hard to identify, but it looks quite similar to other yellow Acarosporas that it co-occurs with. This is a very maritime species, and we found it up sort of at the south end of Big Sur on one of our sandstone sites. And then just one more little pair here, Aspersilia santamonicae and Aspersilia brucei. We both recorded this on our sampling site on sandstone along High Mountain Lookout Road up in Los Padres National Forest. These are both new records for San Luis Obispo County. And they kind of just look like grayish white splotches on rocks. And these are actually probably the ones I might be most excited about, just because Aspersilia is just a really vexing genus for me, really hard to identify, especially in the field. And so to add a couple more to my mental list is really, really helpful. And there just aren't a lot of records at all for either of these species, especially santamonicae. It's really basically only known from some localities in the Santa Monica Mountains, and then I think one location down in Baja California there. And then we've come across 3 species which appear to be undescribed. They're all Caloplacas, or sort of fall into that broad group. One is this one with the gray thallus, a gray kind of lichen body, and then the really reddish fruiting structures. And as far as we can tell, that's a new taxa, and we just need to figure out. We need to confirm that first of all, and then figure out if we can describe it as a new species. And then a couple other ones that my collaborator already had on his radar that showed up in our quadrats were this Caloplaca that Jason has nicknamed Tangerina, as well as Caloplaca, well, something that keys out pretty closely to Marina, but isn't really, and it's just so far away from the right habitat. Caloplaca marina would be a very coastal maritime species, and this is in a really hot, dry inland environments, and it just doesn't quite key out correctly either. So that's all I got for you. Just wanted to show you some of our preliminary findings and some interesting species we've come across so far. A big thank you to the California Native Plant Society, SLO chapter. The support has really gone a long way, and it's not unappreciated. So I really appreciate your support. I'd also like to thank my thesis committee, Dr. Roger Karuna, Jason Dart for sure. I'm not sure if he's here, but so hey, Jason. Alan Friday, Rick A. Naceborg, Scott Johnston, Jen Yost, and Cameron Williams. Everyone has been super helpful, and the list goes on, but I'll stop it there, and then I'll mention Amanda Gersoff, my undergraduate research assistant, who has been awesome. She worked with me last summer. We were supposed to do a bunch of field work. COVID kind of scrapped that plan, and she was really instrumental in helping me start the process of going through this review of lichens of ultramafic substrates in North America, which hopefully will get published down the line. So, all right, thanks everyone, and that's all I got for you.
Very awesome, Michael. I think what I've just learned is that you and Jason are gonna have to lead a lichen hike for us when we're back in action on field trips. I'm scheming. But anyway, a big round of applause, Zoom style. You can't really hear it, but that was super awesome. We're excited to have you as a scholarship recipient. So thank you for that. And thank you for joining us here tonight and sharing all of this. Melissa, I'm sure you saw it. There are 2 questions that popped into the chat. I don't know if you wanted to address those now or.
Well, I was curious about one, Michael. There was one question about whether lichens exhibit seasonality like plants in a grassland do. Is there something? Let's take that one, and then maybe we can put the others off.
Okay, we will address all of them, but yeah.
In other words, does it matter when you go out and sample, like springtime versus summertime, or?
You know, I've thought about that a little bit. I think for my purposes, I've decided that it doesn't really matter because most lichen species are just so slow growing that, you know, when I've returned to lichen quadrat sites, one thing that's actually really helpful to reestablish, okay, where was my quadrat? Because I have various photos that you can use to reestablish it, but what's often really helpful is like the pattern of lichens on the rock that you can use to like just refine the quadrat because they don't have permanent markers on them. And so I've heard the term ephemeral lichens before. I don't know too much about that. But for the most part, they're just so slow growing that it's a very slow pattern. One thing I will mention is when lichens are wetted, their colors often become more or less vibrant. So they change their appearance a lot. And so that's definitely something to take into consideration. Like if it's a moist, dewy, foggy day out, or if it's just rained, that could potentially help you identify species or even hurt you identifying species depending on the lichens in question.
Okay. Shall we move on to our next speaker and then take the rest at the end, Kristen?
Yeah, I like that. I think it's a good flow, but we will hold I see them, we'll hold those questions and we'll get to them. Okay. So Stephanie, if you want to go ahead and co-opt the screen share, I'm going to just start talking and give a little introduction here for Stephanie Calloway, our second of 2 McLeod Scholarship recipients for this year. Stephanie is conducting her research with Dr. Jen Yost, another one of our chapter members here at Cal Poly. Stephanie has a BS in entomology from UC Davis, where she studied the charismatic invertebrates known as water bears. I love this trajectory from water bears to rare island plants, aka tardigrades. And she also had the incredible opportunity to describe a new species of tardigrade from Alaska during her work. She has worked all over California, pursuing careers in entomology, botany, ecology, and recently spent 4 years at the Santa Barbara Botanic Garden as part of the conservation and research team. And during her time there, she was inspired to go back to school and get her master's degree. She plans to continue working in plant conservation in California when she is done with her master's. And fun fact about Stephanie, in her free time, she enjoys observing and collecting pollinators, especially bees, and documenting their interactions with plants. And before I turn it over, I'm just going to say, Stephanie, I second that. I really love watching bees interact with plants. So go ahead and take it away and let us know what you're researching.
Wonderful. Can you guys all hear me okay?
Yes, that sounds good.
Okay, great. All right. Thank you so much for having me, CNPS SLO, and I'm so excited to be here to talk to you all about my research. And I'll be talking to you about the rare Northern Island mallow out on Anacapa Island and its past, present, and future conservation. So a quick outline about my talk. I'm going to first give you all some background about the Northern Island mallow. I'll discuss its history on Anacapa Island, and then I'll talk about past and present conservation, and then jump into the future conservation part that is kind of the focus of my research. The Northern Island mallow is a perennial shrub in the Malvaceae family. It can flower and fruit year-round, but it usually peaks in the late spring, like you see in this photo with these beautiful, vibrant blooms. It can become quite a large shrub. So this plant that you're seeing here is about like 8 feet across and over 6 feet tall. Another name for this plant is called the tree mallow. It has these just amazing flowers. On the left, you can see a flower where the pollen has matured on the filament column. So the pollen matures first. It's incredibly sticky. You can see an STM photograph up in the corner. It has these little protrusions that just make it stick to everything, including that little jumping spider down in the corner. And then the right photo shows the pollen has kind of all gone away, and these beautiful curly stigma have come out of the center of that filament column. And spatially separated timings for these, so the Melva can't pollinate itself. So it has, each branch is kind of just loaded with these fruits in the peak fruiting season. And each plant can produce thousands and thousands of viable fruits. And when they disperse to the ground, they typically, fall as a whole fruit, and they can actually stay on the plant for a really long time until this happens. And then within each of those fruits, there's about 7 seeds. The Northern Island mallow is a part of a group of mallows that has diversified across the California Channel Islands and the Baja California Islands. And the Northern Island mallow goes with the blue circles on the map. Its closest relative is the Southern Island mallow, which you can see looks pretty similar, but that occurs on Catalina and San Clemente Islands. And zooming into those Northern Channel Islands, so these are the Channel Islands off of the coast of Santa Barbara and Ventura. And this is where Northern Island mallow occurs. So unfortunately, as many of you may know, introduced herbivores were introduced to the Northern Channel Islands pretty early on. So we don't really have a good understanding of the distribution of the Northern Island mallow, because everything was pretty degraded by the time we wanted to answer that question. So those areas where we're not really sure that it occurred are represented by the blue empty circles. And the blue solid circles are areas where there's really strong evidence that it occurred there. We know that it occurred on Anacapa and San Miguel Islands. And on San Miguel Island, the Melva is known from about 150 plants, and many of those are recruiting. So that means there are seeds that are germinating, and those seeds are maturing into, they're growing and they're surviving as adults after they germinate. On Anacapa Island, the Northern Island mallow was actually previously extirpated from the island. And luckily, the Park Service and Steve Junak collected seeds from the last remaining plants and used those seeds to grow plants and plant those plants back out on East Anacapa Island. And now there is a large restoration site managed by the National Park Service and their partners, the California Institute for Environmental Studies. And I just want to go over some a little bit more detailed history of Melva on Anacapa Island, because I think it's fun to think about this in that context. So in 1884, Melva was described from Anacapa Island by the botanist Kellogg as being from the island of Anacapa off the coast of Santa Barbara. And then already by 1886, sealers reported to the botanist Lichens. So apparently this was like a common thing that, like, I always read all this stuff where it's like, this botanist talked to these sealers to get information about the Channel Islands botany. But it was reported that although Melva was once abundant, that it was now scarce on Anacapa. And you'll notice it says San Nicolas Island. So little uncertainty there, like I was mentioning with San Nicolas. And that is largely because, like I talked about earlier, they introduced herbivores, and mostly sheep. So you can see a photo on the right. This is a photo of sheep on Santa Cruz Island. I could not find one for Anacapa, but there were certainly thousands of sheep that grazed out there. And although the first ranching lease on Anacapa was in 1902. There had been, like I said, grazing way before that. And by the 1930s, sheep had already destroyed most of the native plants. And that's when in 1930, Ralph Hoffman, the botanist, saw 4 or 5 plants on a steep hillside above the sheep landing on Anacapa Island. In 1978 and 1979, there were just a few plants seen by both Ralph Philbrick and Steve Junak. And then in 1994, it was last seen on Middle Anacapa Island by Steve Junak, and that is when seed was gathered by Steve and Sarah Cheney before those last plants went extinct. So I want to talk a little bit about some past conservation here. Melva was really just one decision away from being extirpated from Anacapa Island forever. And these are the people who decided that it was worth saving. So Steve Junak on the left and Sarah Cheney collected seeds from the last remaining plants. And they gave those seeds to Dieter Wilken at the Santa Barbara Botanic Garden, where he propagated them. And those plants were the plants that eventually returned to East Anacapa Island. I think I need to double check this, but I think it was in the 1990s that they were planted in a few pockets around Anacapa Island. So there's some plants that are quite large now that were reintroduced a while ago. Some of them have died because apparently they are relatively short-lived shrubs. So this was really the beginning of how Melva was just that one decision saved Melva on Anacapa Island. And now those initial plants are what was used to, collect seeds and create the new restored population on East Anacapa Island that I'll talk about now. So, as I mentioned, the National Park Service with the California Institute of Environmental Studies installed a really large restoration planting in the slope below the lighthouse on Anacapa, if anyone has been there. And, sorry, excuse me. Yeah, so they wanted to do this not only to restore or to conserve the Melva, but also to restore habitat, just to increase native plant cover and increase habitat for seabird species. So on the left, you can see what the site looked like in 2016. There's some young Melva that have been initially planted there. And then here's what that same site looked like just 2 years later in 2018. Looks like a totally different site. This plant grows incredibly quickly. It's amazing. You put it into the ground when it's, less than a year old, and they give it a little bit of water, but it really just takes off. And here's just another really beautiful example that Andrew Yamagiwa took from the California Institute for Environmental Studies. They're the group that does all the restoration work out there. And the site just looks totally different than it did before they started the restoration. And you can see they not only planted all this Melva, but there's kind of a diverse array of other coastal sage scrub species. And this beautiful resource appeared really quickly and animals followed. So despite the really short amount of time this plant has been growing on Anacapa again, it's you walk into the Melva planting and it's just a buzz with life compared to other places. And here's just like a smattering of a few of my favorite species that I've observed out there, including the Anna's hummingbird, which they fly up to the flower and just get pollen deposited all over their cute little foreheads. And this is an event that happened this year when I was out collecting data in May. There was a migration of what was probably multiple thousands of warblers coming through the Melva planting to feed on insects within the Melva planting. I'd be collecting data and all of a sudden I'd just be surrounded by like 50 birds. And it was really amazing. So through the amazing efforts of everyone that came before me, the reintroduction of Melva to Anacapa has been a true conservation success story. Like I said, Melva was literally one decision away from not being there anymore. But unfortunately, there are still some issues that need to be addressed for Melva on Anacapa in order for it to continue persisting there in the future. As I mentioned before, land managers have noticed that this plant is not recruiting. So the picture that you're seeing here is one of the only like 2 or 3 known recruits within the Melva planting, despite the fact that each of those plants, there's over 800 plants that have been planted, produce highly viable seeds. And before I jump into the core of my experiment, I first want to talk about the Anacapa deer mouse. So this is Anacapa's only land mammal. It's an endemic species to Anacapa Island, and it has an interesting history itself. So there used to be black rats on the island, and they wanted to eradicate them because they were really harming seabird populations as well as a lot of other species. So they gathered up all the deer mice and lizards on the island, and then they placed rat poison throughout the island. They waited for all the black rats to die and for the poison to become not toxic to the animals they wanted to re-release. And they re-released the deer mice back onto the island. So they're also a special island species, but the land managers out there have noticed that, hey, like these deer mice love eating like all parts of the Melva, including the fruits. Could they be preventing the recruitment of this species? Is this why we're not seeing them growing? So now that we kind of have that piece of the puzzle, I want to talk about what could be going on with Melva by looking at its life history. So let's go through the life history of Melva together. So the seeds ripen and disperse, like I mentioned, and fall to the ground. There doesn't seem to be any issues with dispersal in areas where Melva naturally disperse and germinate. You can see them growing kind of at the edge of their parents. The parents kind of act as a nurse plant. So once they disperse onto the ground, they enter the seed bank, and there can be several things that happen to Melva, the seeds once it enters the seed bank. For example, they can be eaten by the deer mice. Next, germination and establishment. So as I mentioned these seeds are highly viable in the greenhouse. So there can be near 100% germination rates in the greenhouse, but there have never been really any in situ germination experiments. Unfortunately, there's not really any juveniles that are currently living on Anacapa, but we know that when plants are planted in the ground, they're quite young and they seem to grow very well and quickly and can grow into adulthood and transition to that adult life history stage where they become reproductive. They flower prolifically and they produce a prolific number of viable seeds. So we can kind of think about what's happening, like where some issues can be happening with Melva here. And the text and the arrows in black are areas that are hypothesized in the study to be limiting to Melva recruitment, and the areas in gray are where we think Melva is probably doing okay. So you'll notice there's some pre-dispersal seed predation. So that's, are the mice eating the fruits on the plant and how many, and same with post-dispersal seed predation. And also you'll see seedling predation on here. Perhaps they're eating the seedlings once they germinate. So now that we have these hypotheses from these different life history stages of Melva, we can conduct experiments at each of these stages to determine where the greatest amount of seed loss is happening. So I really have 6 main parts to my experiment that you're seeing here. So the first is characterizing the pollinator community. So as Kristen mentioned, I am just a huge nerd when it comes to bees and insects. So I really want to just document as much insect biodiversity on Melva as possible. And there's not much known about it, so why not? Number 2, I want to quantify a seed budget. So how many seeds does a single Melva plant produce? And then once I know that, I can do these little experiments to see where the most seeds are removed. So I can do experiments on the plant through pre-dispersal experiments. I can quantify post-dispersal predation once the seeds disperse into the environment. And then I can also do germination experiments. And number 6, I can quantify the seedling depredation rate by deer mice. And I'm going to go through each of these in a little bit more detail now. Oh, but first, so the overall goal is to kind of have something similar to this, where we can extrapolate to the Melva population, and we can get these percentages of where the greatest propagule loss is happening, and then we can make recommendations to the Park Service and to their partners as to where would be the best life history stage to focus conservation resources. So number 1, pollinators. So I am collecting specimens, and I'm making specimens, and those specimens will be ID'd to the best of my ability and deposited into museums. And aside from specimens, I also have an iNaturalist project. And if you guys are familiar with how to scan a QR code, you can go ahead and do that. And that's a link to my iNaturalist project, where you can see I've observed over 70 different species, not just me. Anyone can add observations to this project. And these are over 200 observations. And then, sorry, that video looks kind of funky now. But that video you saw was this andrinid bee just drunk on Melva pollen, and I just love that video. So I wanted to share it with you. And then another cool thing to point out, you'll see on the right side here, the Buckwheat Masked Bee. We caught that bee on a Melva, and I put it on iNaturalist, where one of the world's bee experts, John Asher, he like goes through all my bees instantly when I put them on there. And for this one, he claims that this is a potentially new island record of bee species for Anacapa Island. And I don't feel as proud after hearing about Michael's like 100 new species of lichen he's discovered in SLO County, but I was pretty proud of this. Okay, and we'll move on to the second part of my experiment, seed production. So the point of this, like I said, is really to create a seed budget per plant, so we know how many seeds are developing on each Melva. So across 20 different randomly selected Melva, I counted all the fruits, flowers, and buds on every Melva. And I did that by just spending a lot of time with a clicker, and it took a really long time per plant, but I got counts across those 20 plants. Next, the third part of my experiment, looking at predation on the plant of buds, flowers, and fruits. So now that I have counts of all those buds, flowers, and fruits, the idea here is I can look at how many of those transition to the next life history stage. So how many buds go to flowers, how many flowers go to fruits, and how many fruits transition to ripe fruits that disperse. And I can do this in both deer mouse included and excluded treatments. And to exclude deer mice, I actually just use these little organza bags. If you make it just hard enough for the deer mice to not get in, they usually don't, because there's just so much other food availability that they don't bother with the organza bags. But across 20 plants, the same 20 plants that I did the counts on, I bagged 10 fruits, 10 flowers, and 10 buds, and then, like I said, bagged half and left half open. So this totaled in 600 total floral units across my replicates. And I meant to say that a lot of I did some preliminary experiments last year, but the core of this work has been conducted through me being out there for 20 total days in the month of May. So a lot of this is kind of just what I've done and not necessarily results yet, but I will be able to share preliminary results from one experiment. But just to kind of set the scene, these experiments were done in May. And to kind of look at deer mouse predation on each of those plants, this is kind of what it looks like. So it's really obvious when a deer has been chowing down on a plant like this. They have these really distinct gnaw marks where they remove the fruit from the pedestal. And as you can see, they eat the buds, the flowers, and they also will even gnaw into the pithy center of the melva. So number 4, post-dispersal seed predation. The idea here is that I am measuring how many seeds are removed from the seed bank once they have dispersed. So across 20 different randomly selected locations, I put out 20 fruits at each of those spots on 3 different nights. And I would do that in the evening, and then I would return the next morning to see how many fruits were removed. And from that video that you just saw there, that was just really one deer mouse that removed about, I think it removed about 50 fruit over the course of 4 hours or something like that. Looks like I didn't put my number on this, but this is number 5 in that diagram I showed you. So looking at germination and mortality of Melva in the field. So across 30 different plants that were randomly selected, I created little 30 by 30 centimeter plots and sowed 15 seeds per each of those plots and assigned them to 3 different treatments: caged, open, and controlled treatments. And this totaled in 1,350 seeds that were sown throughout the planting. And I did this in November of 2020, before the winter rains could come. And I placed those around the 30 plants. Maybe you're wondering about that, because this is, like I mentioned, where we see Melva in areas where they are recruiting germinating. So they really like to be kind of like at the edge of the adult plants, because the mother plant kind of acts as a nurse plant. You can see this wonderful fog drip underneath the adult plant. That's all from fog drip that accumulated in the summer months. So it's just a place where the seeds like to grow. I figured I'd get a little extra water from my experiment, since it was a drought year especially. But despite it being a drought year, I got some germination in my plots. So you'll see this is just some preliminary data, but in my cage treatments, that's the only treatment where I got any germination. And on average, there was about 1 thing that germinated per plant. And I didn't get any germination in the control or open treatments, and deer mice were definitely removing them from those. You could often go visit the very next day that you sowed those seeds and see that all those seeds had been like removed in a perfect grid. They had just been picked out by deer mice. So overall, I got 40 germinants total across the planting, and so far, unfortunately, 15 have died. I think this is because it's been a drought year and they just haven't had enough water to persist. And then finally, the kind of the last part of my experiment is looking at seedling predation. And I wanted to get out and do similar experiments as I did for the seed removal trials, where I put them out and see how many were eaten. But unfortunately, COVID kind of interrupted some boating schedules in the winter, and it was really just a rough season for the seas to get out there. So a lot of my trips were canceled this winter. So instead of doing that part of the experiment, when I was able to get back out there in May, I decided to pull the cages on about half of my seedlings that had germinated and just see what happens as the deer mice are allowed to potentially eat them. So you can see on the left, there's some that are doing really well. This was a seed that just germinated in November. It's already getting quite tall and woody at the base. The seedling in the middle, that is one that has been munched on by a deer mouse. And the one on the right, there was a pretty large wind event that came through and killed some more seedlings. It just desiccated a lot. So this is an ongoing experiment right now. I'm continuing to do checks on these as I do more visits out to the island. So thank you guys for that. That was a lot to take in. I have a lot going on out there, but I hope that you guys enjoyed that talk. Thank you all so much for being here. And I just want to acknowledge a few people that have really made my life easier. So the National Park Service, especially Dave Mazurkowitz and Annie Little, who really helped me get my permits. The California Institute for Environmental Studies, Andrew Yamagiba, who's here tonight, has been just an invaluable resource. He's like one of the main men responsible for installing that beautiful restoration. Elaine Matson for really helping me check on my experiments when all my boats were canceled. And of course, California Native Plant Society SLO chapter, thank you guys so much for the scholarship. It's been so helpful to have that funding from you guys, and also from the Channel Islands chapter. Getting out to the islands can be expensive. Southern California Botanists, Botanical Society of America, and of course, my wonderful advisor, Jen Yost, and Matt Ritter, Matt Williams, and Kristen Hausenstaub-Lehman, and Adam Searcy, who has been my unpaid field assistant for the entirety of my project. And with that, do you all have any questions?
That was fantastic, Stephanie. I really hate to be repetitive, but every time at the end of a talk, I just hate that you can't hear the applause. So I say it every time because that was awesome. Some people have turned on their cameras. I see that your advisor, a couple of your advisors are here. Hi, Matt. So yeah, I'm seeing
Hi,
the
Kristen.
Yeah, nice job. That is a lot of work. That's incredible. And you know, maybe when you have some additional results, I think this group would be interested to hear what you're finding. So that's incredible.
I've got all of my data from May, and I just wish I had time to look at it a little before the talk, but next time.
Just blame COVID. Just blanket blame of COVID. No, that's great. Okay, well, that was wonderful. Thank you so much to both of our speakers tonight. Really incredible work going on here at Cal Poly. I'm very impressed with both of you, with everything that you're doing. New county records, new island records, species, everything. That was awesome.
Yes, very, very nice. Very good.
Yeah. Matt, were you going to chime in, or are you just you're just here for
That was
support?
great. Those are great talks, and thank you guys for continuing to support our students. This is awesome.
You bet.
Yeah.
You bet.
It is incredible. So I think maybe the simplest thing to do will be to start with the oldest questions in the chat so we don't miss them and move forward, and then we can just go from there. And the oldest question in the chat that hasn't been addressed yet came from Dave Chipping, and it's directed at Michael. If you want to go ahead and unmute yourself, Michael, he asks, and you can see it, but I'll just read it for everyone's edification here. Are the fungal and algal components, rather than the whole composite lichen, showing the same distribution trends as what you were talking about with your observations in your study?
Yeah, that's a great question. So interestingly, with lichens, the fungal component of lichens is usually only found as part of that lichen symbiosis, and so you won't find it sort of free living. So the fungal distribution is the same as the lichen distribution, basically. For the algal components, a lot of times for a lot of these lichen species, it's just they've identified it to the genus of, for example, alga, and they haven't gotten further than that. It's like, oh, this is a trentipolioid algal photobiont or something like that. And you know, some fungi might be able to utilize multiple species of algae. So short answer is I have no idea when it comes to the algae distribution, but that would be a really interesting thing to look at, and someone probably has, but I don't have too much to add on that aside from that.
That was a great response. I was wondering if that can even be known, but that was great. I'm scrolling here, some nice accolades and thanks for your presentation as well. Hopefully you saw that. Oh, Marlon Harms, I saw this question come in and I wanted to make sure we didn't miss it. Marlon Harms, who, before I read his question here, you should know he's an incredible photographer. This is for Michael again, incredible photographer, member of our chapter for a long time. And he asks, would you be willing to receive some photos that he has and identify lichens for him?
I would love to try. And, you know, if I can't, I maybe know some people who could potentially, if they have the time. So, or at least point in the right direction.
Yeah, maybe if you're willing, you can post your email in the chat, or I can just connect you to after this if you prefer to do that.
Yeah, for sure.
Let's see, Melissa, if you see one that I'm missing, feel free to jump in.
You're doing great.
I'm just scrolling through here.
You're on a roll.
And you know what? We have our meeting format here with about 40 participants. So if someone does want to jump in and just throw out a question while I'm scrolling, feel free.
There's one from Paul Escoffier. I'm not sure if I'm pronouncing that right. For Stephanie, is it possible that the original natural occurrences of Melva were in locations inaccessible to the mice? Switching from Michael to Stephanie. That was a quick segue.
Thanks for the question. Yeah, that's an interesting one to think about. I think it's definitely possible, kind of like the issue with Melva, knowing about what the natural occurrence was like. We probably have the same questions for deer mice. We don't really know much about their life history besides or before everything got really messed up. But what I do know on Anacapa Island, the deer mouse densities on, I think, Anacapa Island and Santa Barbara Island are, they're the most dense anywhere in North America, because there are no top predators there essentially for them except for owls. And other birds of prey that may be eating them, but there's no land predators for the deer mice. And it's interesting because on San Miguel Island, where the Melva are naturally recruiting, there are island foxes out there. So perhaps, you know, the island foxes play a role in keeping the deer mice populations down a little bit more. But yeah, I mean, I could definitely see what you're talking about, Paul, like if it was like on a cliffside on some of those steep rocky terrain places on Anacapa, it's possible that maybe mice just would not want to go there. But I don't know that for sure. Oh, we've got Andrew chimed in here. Oh, yeah, Andrew, the restoration biologist out there said, my experience is that mice can access most anywhere.
Nice. Yeah. Well, good question and good answer as well. Marlon, hopefully you saw Michael posted his email in the chat there. You can grab that. And someone, I don't know if you saw that latest step for Stephanie, has the lack of predators for mice on Anacapa always been a challenge for Melva. Was that part of the last question? No. It's real quiet.
No,
Am I on mute?
I have a possible thought on the island foxes. During when there were sheep grazing, were there, of course we don't know, were there island foxes on Anacapa prior to the sheep grazing that would have kept the deer mouse population down?
You know,
And that they may have gotten extirpated or just crowded out with the grazing.
yeah, you guys are really going to challenge my knowledge of like geological history of the islands right now, because so technically, you know, all of the northern islands used to be connected and they used to be one island. So at that time, Melva definitely occurred on all the islands because they were the same island. And I'm sure the fox introduction happened after. So there, to my knowledge, has never been foxes on Anacapa Island.
Nice.
But yeah, okay. We kind of skipped over the Haas question, but I guess that's related to the mice also. David,
Yeah.
David Chipping is raising his hand. Go ahead and unmute yourself, David.
While the fox population was being discussed, I looked it up. There never were foxes on the island because there's no water.
Okay. We're getting a full breath here from lichens to plants to island mammals. I love it. And I don't know a lot about any of it, so.
Any other questions from anybody? Anybody want to chime in on any other Kristen, did you see something else in the chat?
Just a couple comments from Cindy that sort of posed as a question that pollination seems very periodical on the islands. And a follow-up that question, are you posting your pollination photos on INAT or elsewhere? And I think you said your INAT is where you're posting all that, right?
Correct. I am collecting specimens, and I hope to I will deposit those into museums, but I'm also taking photos, and I have an iNaturalist project where I'm putting all those photos on, and it's really amazing that I have this like world bee expert that just likes to get on there and ID all my stuff.
That's the beauty of INAT.
I love INAT so much. And then about the periodic nature of pollination. So one thing I've really noticed from spending so much time out on Anacapa is that it seems like the pollinator community like is really active for a pretty long time out there. Like I was out there, you know, last fall, and I was still, there's some really late bees that were just still doing their thing. And the pollinator community is super healthy on Anacapa Island. Like you look at one plant, and I was observing one plant, you know, a few weeks ago, and I counted like there were at least 8 species of bees visiting this one species of plant. And I don't know if that's because the islands are just more isolated from things like pesticides, or whether there's some other part of the habitat that just like makes it very good for bees out there. yeah, it's an amazing diversity of pollinators out there. I've been really pleasantly surprised.
That's fantastic to hear. And an interesting comment about the pesticides. Sounds like somebody has a PhD question to address. Yeah, and actually that ties a little bit into the last question that just popped in the chat from Lindy Althouse, for both of you regarding what taxa or ecological phenomena maybe. I'm sort of amending her question because I'm curious. Are you interested in studying next? Really putting you on the spot there with that one. Oh, you're on mute, Stephanie.
Oh, I was just rereading the question.
Oh, yeah.
Oh, man, that is a real, I don't know if I could pick just one. I mean, I love bees and plants, and I'm probably just gonna continue on with those as like my labors of love.
Awesome. Do you have anything to add, Michael?
You know, I think I'm gonna stick with lichens for a while, but I think lichens of like soil crusts and how they interact with plant communities is really interesting. And I think, I don't know too much about this, but I've been curious about like how they could potentially be used in restoration projects. So yeah, those are some things I've been floating around in my head, but yeah, I don't know. So many cool things out there.
All right. Sounds good.
Very cool. Well, not seeing any more questions floating into the chat or any more hands raised.
Okay.
This one?
I have if you got time for the little lichen story about Freddie Fungus, who was a fun guy, and Alice Algae was all well, took a liking to each other. Alice's mother disapproved because they were living in symbiosis, but they had a good working relationship. Freddie builds a house and does the shopping. Alice does the cooking. Now, when they first got together, they went out on a limb. But rumor has it their relationship is going on the rocks. Some people try to say their marriage is on the rocks, but they're not married. They live in symbiosis. Does Michael have any more little quips to add to the Freddie Fungus and Alice Algae story?
You broke up a little bit there, but I think I got the gist of it. That is the most complete version I've ever heard. So I have nothing to add. That is wonderful.
Very punny, very punny.
And on that note, I think.
Right.
Real quick, Stephanie dropped a link to her INAT in the chat.
Rocks. Yeah, it's funny, right?
Yeah, so if everyone saw that and you wanted to check out Stephanie's iNaturalist observations, she dropped a link in the chat before you sign off. So thanks for doing that, Stephanie. And yeah, I think just a thank you to both of you. Some applause coming through, and keep up the good work. Send us some updates when you have them. We're interested, and it's really, really, really awesome work that you two are doing.
Right. And thank you very much. And yes,
great job, you guys.
Yep.
Thank you, and thanks to all of you. It was a joy.
Yeah, thanks for coming, everyone.
Great presentation.
thanks to all of our loyal members for showing up tonight. Appreciate it all. All right. Good evening to you all.
Are we going to have any summer meetings?
No summer meetings. We'll see you in the fall.
Thanks, everyone. Thanks, Stephanie. Thanks, Michael. Have a good night.
You too.
Hike soon.
Yeah.
All right.
Okie doke. All right. Good night, all. Good night, Charlie.